We are. All right. So we're beginning with this peptide comprised of, let's see, 2, 4, 6, 8, 10, 12, 14 amino acids. This was made, it's a synthetic molecule, synthetic peptide made in the early 2000s. Professor Marcy Waters at the University of North Carolina, Chapel Hill, actually came through the University of Wisconsin when I was there and gave a talk on this. It's pretty cool stuff. But this molecule forms the smallest increment of a beta sheet. And for our purposes, when we're talking about beta sheets, that is tertiary structure. And a beta sheet is composed of a beta strand, that's the Greek letter beta, plus a beta turn, plus another beta strand. And anti-parallel sheets have this basic structure. Here is, we'll use this sort of strange arrow shape to indicate the strand. Then there's the turn, and then there's the other strand. And the arrow the arrow points towards the C terminus. So this molecule is called an anti-parallel beta sheet or beta hairpin because you've got one beta strand going along from N to C, then there's a quick 180, and then the second beta strand going along from N to C. So here's the sequence. A couple of things in the instructions to help you out. This AC symbol there stands for an acetyl group. So the AC at the N-terminal side of the sequence means that the N-terminal amino acid in your sequence, whatever it is, the ammonium group is not actually charged. It's tied up in an amide bond with an acetyl group. So this portion here is the AC group, and this is an amide bond. So when you see an AC on a sequence written out like that, that just means you've got this, I'll put it there, acetyl group at the N-terminus, which means the N-terminus is not charged. There's sometimes reasons why you want that to be. It's a minor issue, not terribly important, but since we're going to draw the structure, I had to explain it. Then the NH2 at the end of the sequence refers to the fact that the C-terminal amino acid is not charged as the carboxylate, but the C-terminal carboxylate is actually also forming an amide bond with an NH2 group. And acetylation and amidation are what those things are called. And they make the N-terminal group and the C-terminal group both neutral. You can see for the C-terminal amide, there's no negative charge. And for the N-terminal acetyl group, there's also no negative charge. This is something you do sometimes synthetically to peptides when you don't want there to be charges at the end groups. That's not terribly significant. Since we're going to be drawing the molecule, you need to follow along. Another important thing that'll help you out is to realize that these cysteines at either end, cysteine 1 and then cysteine 14, are going to be disulfide bonded to each other. So in our structure that we draw of this molecule, those two cysteines need to end up close to each other. If we're going to sort of adopt the cartoon version of this peptide, what we're going to expect to see is here is the N-terminal cysteine with its acetyl group. Gave myself need more space. And then here is the turn. and then here is the C-terminal cysteine with its carboxamide group, or the amide group on the C terminus. And you expect these two cysteines to be disulfide bonded to each other. So that's one feature, just looking at this sequence, the information I'm giving you as we're going to draw it. That's one piece of the puzzle. And one of the things that holds this molecule together in a hairpin is the fact that cysteine 1 and cysteine 14 are forcibly covalently linked to each other. They can't get apart. And that helps constrain this molecule to be in this beta hairpin conformation. Another thing you're going to notice is along the way, specific amino acid residues are going to be hydrogen bound to each other across the strands, across each beta strand. So we'll call these cross-strand hydrogen bonds. Okay, so just to be absolutely clear, this portion in pink represents what I'm going to call the N-terminal beta strand. This portion in green, we'll call the C-terminal beta strand. And then the segment in between them we'll just call a beta turn. And mapped onto the sequence here, you wouldn't necessarily know this from the sequence, nor would I expect you to look at that sequence and say, oh yeah, it's a beta hairpin. But if I told you that it is, then I'd like you to be able to draw it and visualize the structure. So everything from this cysteine all the way up through the valine is the N-terminal beta strand. And then everything from this O all the way up through the C is the C-terminal beta strand. Oh, right, I need to talk about what the O is. We'll get to that actually as we're drawing the sequence out. O is an unnatural amino acid that was used by Professor Waters for a lot of reasons that aren't necessarily important today. Let's see, and then lastly, this portion, I need a different color, don't I? What haven't I used? Well, gray. This portion is N and G in the middle are the beta turn. Yes?
Does it specify that somewhere in the directions, or is that just something that you're telling me?
In the directions up here, it tells you the first 6 residues are the N-terminal beta strand, residues N and G are the turn, and the last 6 residues are the C-terminal beta strand. Yeah?
Didn't you say that G is usually part of the turn now?
G is frequently part of a turn because it's more flexible, and the peptide chain has to twist back on itself at that portion. A whole interesting area of chemistry is related to which amino acids go best in the turn. This particular combination, asparagine and glycine, are particularly good for reasons that we could talk about if we had forever, but we don't. So, follow up? Yeah.
Well, this is a separate question. Will 2 cysteines on either side of the bend always do a sulfide bond if they're like parallel to each other, or?
Yeah, so you will see when we draw this beta strand, the question is, will 2 cysteines on either end, one strand versus the other, will they always form disulfide bonds? Do they have to line up parallel to each other? That's a complicated question. The answer is you're going to see in this sequence that they do line up with each other. It would be possible to place cysteines in a relationship where they don't line up with each other, and that might actually cause some problems for the structure. Yeah, I saw your hand.
Will the turn always have 2 amino acids?
Great question. Will the turn always have 2 amino acids? There's multiple kinds of turns. There's like a whole paper that was written maybe 20 years ago called the Anatomy and Taxonomy of Beta Turns, I think is what it's called, or maybe it was protein structure in general. they actually use data to classify different kinds of beta turns? The answer is no. You could imagine putting a lot more amino acids in a turn. What you're going to see is the thing that defines a beta turn is you have 2 amino acids and then in the turn, and then the 2 amino acids that flank that turn are going to be hydrogen bound to each other. So that's the definition of a beta turn. 2 hydrogen bound amino acids and then a 2 amino acid bridge. That's what we're going to stick with since it's easiest to draw and reason your way through. But no, nature's not so limited. All right, others? What I've sort of outlined here is called a ribbon diagram, and this is frequently the way people visualize protein structures, because when protein structures get big enough, if you look at all the atoms, it can be overwhelming. And so what they do is sort of illustrate the main features of what's going on in the molecule with a cartoon. Okay, so to draw this, what I'm going to recommend is that we start here with the 2 amino acids in the turn, and then the 2 amino acids that flank that turn. So, and we're going to start over to the the right-hand side of our paper. We're going to put our turn here, and then ultimately one strand's going to be here, and the other strand's going to be there. So to begin, let's draw the valine that's right here, and I'll number the sequence maybe 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. All right. So we're going to start with valine 6, and we're going to draw it this way, with the carbonyl on the right and the nitrogen on the left. We'll put the alpha carbon in between the two at sort of a peak position, and the valine side chain, in order to have correct stereochemistry, should be a wedge. And then valine looks like a V. Valine's the easiest to remember because its side chain looks like a V. Okay, then down here, immediately below valine, you're going to draw a dotted line. You're going to draw an H and an N here, the alpha carbon in between, and then the carbonyl oxygen there. And you want to draw it in such a way that the amino acid that we're just drawing is hydrogen bound to the valine where we just were. What we're drawing here, this is valine 6, and we're currently drawing ornithine 9. So O equals ornithine. It turns out it is a natural amino acid. It's used biologically to synthesize the amino acid arginine, though ornithine itself is never found in proteins. Nevertheless, the people who were making this molecule used it for whatever reason. So at this position, the ornithine side chain, in order to have the correct S configuration, should be a wedge. And then ornithine is like lysine, but it's 1 carbon shorter. So for ornithine, we're going to go alpha, beta, gamma, delta, and on the delta carbon, we're going to put the ammonium group. Whereas if this were lysine, oh, I did it wrong, didn't I? Yeah, how about that? So ornithine is 1 CH2 shorter than lysine. The ammonium group is on the delta carbon. All right, fair enough. So far we've just drawn valine 6. We haven't drawn anything that's bonded to valine 6, and we've also drawn ornithine 9. We haven't drawn anything that's bonded to ornithine 9. Now we need to do the the two amino acid bridge between the two of them. Any questions so far about what we've done? Okay. These dotted lines, I guess I should have used the color blue, since in my diagram I used the color blue for cross-strand hydrogen bonds. So style, blue, boom. Okay. Now, the next amino acid is asparagine. Its amino group is here. We're gonna go here, a zag down to the lower right, and then here, we're gonna put the alpha carbon, of course, is here, and to have the right stereochemical configuration, I haven't drawn the carbonyl carbon yet, but to have the right stereochemical configuration, the asparagine side chain should be a wedge. Then, at this point, our two-dimensional drawing of a three-dimensional structure is gonna fail. What we really need to draw is the carbon-oxygen pi bond here, sort of pointing straight out at us. We could use dashes or wedges for that, but we're not going to. What I'm gonna do is simply just draw it like that, and then draw the next bond to the amino group of lysine like that. So when you see that, the amino group of glycine like that. So when we draw a beta turn, you have to imagine the bond, the amide bond between the first amino acid of the turn and the second amino acid of a turn sort of sitting on edge. Kind of have to be creative. Then, we'll draw the bond to the glycine alpha carbon, then the bond to the carbonyl carbon, and then the bond to the next. Okay, so this is amino acid 7. This is amino acid 8. Of course, because amino acid 8 is glycine, there is no side chain. I'm showing you the two hydrogens there just to illustrate. And it looks like I've left off H on the asparagine residue. So this is our V, N, G, and then the one letter symbol for ornithine is O. So that's a beta turn. We would call these two amino acids here the turn segment. And I'm just highlighting the backbone atoms. And then we've drawn amino acids 6 and 9, which are the 6 is the last residue in the pink beta strand, and then 9 is the first residue in the green beta strand. Yeah?
Is that a curiosity for the asparagine? Does it matter if the bottom one was there or basically?
Right, so I drew it with the oxygen here and the NH2 there. You can rotate freely around carbon-carbon sigma bonds, so no, that could be spinning around in solution and you could draw it either way. Yeah?
Could you box the letters for the amino acids?
Oh, sure. Make it a little bit clearer. Maybe I should choose a different color, but. Okay, other questions? Yeah.
It's connected by an dash.
Sorry, where?
Like for the two amino acids of N and G, the connection.
Yes. Yeah, the connection between N and G, like the connection between any amino acid in the peptide, is going to be an amide bond. So that's an amide bond right there. We're just not drawing it out in a full zigzag form, so it looks different. All right, what else? Yeah.
The O one, why is it a wedge and not a dash?
It is now a wedge instead of a dash because nitrogen's on the right and carbonyl carbon's on the left. Normally when we've drawn amino acids, we draw them with the nitrogen on the left and the carboxylate on the right, and when the alpha carbon's at a valley instead of a peak, it's a dash. So yeah, okay, good. Other questions? So that was the hard part. All you got to do from here on out is to draw the pink beta strand backwards, starting from position 6. So we're going to go 5, 4, 3, 2, 1. And then we'll draw the green beta strand, starting with amino acid 10, 11, 12, 13, 14. Along the way, we'll have to check ourselves because we got to make sure the amino acids on the top strand take about the same amount of space as the amino acids in the bottom strand. But if you're careful, that's not too hard. So going back from 6 to 5, that amino acid is glutamate. So we can draw carbonyl carbon, alpha carbon, amide nitrogen. For this amino acid 5, the side chain should be a dash or a wedge. So we have right stereochemistry, it should be a dash. And the side chain is glutamate, so there's the beta carbon, alpha, beta, gamma, and then on the delta carbon, you've got a negative charge. And I'm going to need to make a little space for that glutamate side chain. You can sort of do this how you want. I'm going to need to draw it smaller than that. Beta, gamma, delta. There we go. Let's just do it one strand at a time. Now we'll draw the amino acid 4, which is a valine. Here's the carbonyl oxygen of that valine. Here's the alpha carbon, which should be a wedge, and there's the valine side chain, and then here is the amide nitrogen. So this is E and V. Then we'll draw the next amino acid after that. That's a W? Tryptophan, right. Which you know I struggle to draw. And how's that gonna go? I think we're gonna see I need more space for that. So you can draw it however you like. I'm just gonna try again. Tryptophan side chain. Then the next amino acid is R, which is whose favorite amino acid? Pirates, alpha, beta, gamma, delta, and then arginine has this weird guanidinium functional group. It's positively charged at physiological pH. Looks like I didn't leave myself quite enough room for stuff, and so to avoid confusion, maybe I'll just drag that down a little bit. Make the arginine side chain a little bit bigger. And then lastly, we have the N-terminal cysteine, which I'm going to just temporarily draw like this, and then we'll have to form the disulfide bond. And then lastly, you've got an acetyl group. Okay, any questions so far?
Why is it like that?
The AC represents an acetyl group, meaning the N-terminal, instead of being the NH3 plus group, it's tied up in an amide with this portion of the molecule. One of the things that you might notice about the beta strand, which we can now highlight in pink, oops, that's not pink, is that the side chains alternate sides, right? You see how it switches wedge, dash, wedge, dash, wedge, dash. So every other side chain is on the same side of the beta strand. Yeah.
So are the side chains also participating in hydrogen bonds, or is that only the carbonyl like oxygens and the?
Yeah, great question. What are the side chains doing? For the interstrand hydrogen bonds, we're going to focus on the backbone amide nitrogen and its proton and the backbone carbonyl oxygen. But an important question is, are the side chains interacting with each other? And if so, how? The answer is almost certainly yes, and once we finish drawing it, we'll hypothesize some things. Okay, other questions? Yeah.
I wanted to make sure the sulfide bond would have to recalculate to make sure that that was going down. So that
Right. If you want to make the disulfide bond, or if you want it to fold into this structure, you need the cysteines to line up so they're both on the same side of the strand. If you put a cysteine on the wrong side of the strand, it turns out that cysteine oxidation is really easy to do. Even in air, it happens quite quickly. So likely the two cysteines that you've described would still disulfide bond to each other, but doing so might disrupt the rest of the structure. What you're talking about, I would call that having the two cysteines in register versus out of register. Are they at the same spot or are they not? Okay, good. Others? So let's just keep going on that strand. Again, you want to keep each strand taking about the same space. So the next amino acid from 9 is 10, which is also an arginine. Oops. So here is the amide nitrogen of arginine. It looks like maybe I need to draw that bond just a little bit shorter. A hint is try to keep the ends on the bottom lined up with the carbonyls on the top and vice versa. So the alpha carbon of arginine 10 is here. And this is why we're going to sort of struggle with space. In fact, maybe I better redraw my glutamate, alpha, beta, gamma, delta, alpha, beta, gamma, delta. There we go. Draw my glutamate there, and then I'll put my arginine here, alpha, beta, gamma, delta. I'm having to draw the arginine side chain kind of small. And that's okay. We kind of tend to run out of space sometimes. So sometimes you got to draw the side chain small. Understand though that in terms of 3D space, those side chains are going down into the page, so there's plenty of space for them. So here's a time where you could hypothesize a potential cross-strand interaction. What do you notice about these two amino acids, 5 and 10? They're charged and they're oppositely charged, right? And they're pointing towards each other, but downward into the plane of the page. So you might expect there to be a cross-strand interaction there. You might expect, remember back from when we talked about non-covalent interactions in like the second week of class, you might expect a ion-ion pair, an attractive plus-minus interaction there. And there's evidence for that kind of thing going on. All right, let's continue. We need to draw the next NH, and this NH needs to be hydrogen bound with the oxygen across from it. Then the alpha carbon would be here, and the carbonyl carbon would be here. And again, you want that carbonyl oxygen to line up with the NH. The side chain here should be a wedge, and that amino acid is isoleucine. All right, so that's O. then R, then I, and that's amino acid 11. Then we'll have a leucine amino acid. That's 12. Oops, looks like I put the H on the wrong side. That's L. Then lastly, Q. Which should be a wedge. Beta, gamma, delta. This is glutamine. And then finally, the cysteine. Which I'm going to put disulfide bound to the other cysteine like that. So 1, 2, 3, 4, and so on. Nine, 10, 11, 12, 13, 14. And then you can see, and this is one reason why they put that amide group and acetyl group on this molecule, is so you could have one last hydrogen bond. And that's the green beta turn. Or that's the green beta strand. Yeah.
How do you know when you're supposed to make a hydrogen bond?
Great question. How do you know when you're supposed to make a hydrogen bond? You make a hydrogen bond every time. So first, the way to do this is you start like we did with the two amino acids that are in the turn, and then the 2 amino acids that flank the turn. Those 2 that flank the turn, on the top you put the carbonyl oxygen pointing downward, and then you put the amide proton pointing downward. For the one on the bottom, you put the amide nitrogen pointing upward and the carbonyl oxygen pointing upward. Notice that for V valine 6, its carbonyl oxygen is hydrogen bound to the amide nitrogen from ornithine 9. So they're anti-parallel to each other. And their amides are hydrogen bound to each other's carbonyls. We call that a hydrogen bound pair. And that would be like valine 6 and ornithine 9. ORN is the 3-letter abbreviation for ornithine. Notice that for the next amino acid down the hairpin, glutamate 5 and arginine 5, notice that their COs and NHs are pointed outward, away from the other strand. So those are not hydrogen bound with each other. So we call glutamate 10 and arginine 10 a non-hydrogen bound pair. And you'll see that in a beta turn, you have this alternating hydrogen bound pair with non-hydrogen bound pair. Hydrogen bound pair, non-hydrogen bound pair. Hydrogen bound pair, non-hydrogen bound pair. One interesting feature of the beta turn is that in a hydrogen bound pair, the 2 side chains are pointing away from the space between the turn, and they're both on the same side. And then in a non-hydrogen bound pair, the 2 side chains are pointing up towards the opposite side, but now towards each other. So it's thought that amino acids at a non-hydrogen bound pair position actually approach each other closely. Okay, other questions about your beta hairpin? Go ahead.
No, this is unrelated. I was
Yeah.
just wondering what this protein is. What-
So this is actually a model protein. It doesn't have a function, but they designed the sequence to adopt this structure, and it did. One other interesting thing about this sequence is we've drawn it out as though it's perfectly flat, if you were to see a three-dimensional structure, it would be twisted. It would be this ladder-like structure, but then the end on this side, the turn, and then the disulfide bond are going to be twisted out of plane with each other. And what that means is some of the side chains at adjacent non-hydrogen bound positions are actually close to each other. So this is not important at all for this class, but the group that made this was interested in whether this tryptophan side chain could interact with this arginine positively charged side chain. Looks like they're far apart because of the twist. They're actually close together. And they were studying the strength of that interaction using this as a model system. Yeah?
Is the first protein and the last protein always going to be connected, or is it just in this case the start and end?
Yeah, is the first and the last amino acid always going to be connected like that, or is that exclusively just because they happen to be cysteines? Well, it turns out if you don't have that connection there, then the two strands are free to wiggle apart. And what that means is it's okay, but if you force them to be close together, then the molecule is more stable and adopts a more stable structure. It turns out this, in the study that I was talking about by Tatko and Waters, this was their control fully folded compound. They had made it so that the two ends of the strands were forced to be close together and it would adopt this structure. That was an important reference compound for them. I feel like we're about to do the eensy spider or whatever, but that was an important reference compound for them. They made other changes to the molecule without that disulfide bond, and then they were able to reference that to the fully folded molecule and get some interesting information. So there's not always going to be cysteines there. It's a little bit helpful because it helps you know if there are cysteines there, and if your turn structure is right, the two cysteines have to be lined up with each other. Other questions? Yeah?
Will this be on the test that starts tomorrow?
Will this be on the test that starts tomorrow? That was very direct. Some items from this quiz will be on the test tomorrow. Knowing the amino acids is certainly on the test tomorrow. On a multiple choice exam, you're not, like, how could I make you draw a beta hairpin? I could make you recognize one. I could ask you some questions about the structure based on our discussion today. But yeah, one unfortunate consequence of, on Dr. Savage's exams, he'll literally write a sequence and say, draw this in a beta hairpin. Kind of like your problem set does. For a multiple choice exam, I can't do that. And I'm trying to figure out how to do that. I will have that figured out by the final. I don't quite have it figured out today. I think that maybe answers your question. But don't jettison this knowledge permanently. Yeah?
Will you explain one more time why N is wedged?
Right. Why is N, the N in the turn, wedged? Well, first, let's just prove that it's got the right stereochemistry. The stereochemistry for all the amino acids except cysteine has to be S at the alpha carbon. Nitrogen is priority group 1, carbonyl carbon 2, side chain beta carbon is 3. 1 to 2, 2 to 3, 3 back to 1. That is counterclockwise with the proton at the alpha carbon down, so that's S. You follow that?
Are you going to put a random R configuration?
Will I put a random R configuration in there? What do you mean?
Are you going to try and trick us with a question?
So will I try and trick you, not necessarily related to a beta turn, but will I deceptively use the wrong stereochemistry of amino acids when asking which of the following molecules is this sequence? Oh, absolutely. Here's how you do it. Here's how you make sure you know what's right. So, as I've mentioned before, if you draw an amino acid, whether it's on its own or in a protein, here the two squiggly lines indicate the amino acid could just be on its own or it could be in a peptide. You have 3 atoms, nitrogen, alpha carbon, carbonyl carbon. When you draw it with the nitrogen on the left, the carbonyl on the right, and the alpha carbon at a valley in between nitrogen, the side chain is a dash. If you pancake flip that thing upward, so that means bring your spatula under here this way and flip it up that direction. That should change things, so the carbonyl carbon is still on the right, the nitrogen's still on the left, but the alpha carbon's now at a peak, and it should be a wedge. Similarly, if I pancake flip the amino acid this way, now the carbonyl carbon's on the left and the amide nitrogen's on the right, the side chain should be a wedge. And if I pancake flip this bad boy up that direction, then I will have this, oops, and it should look like that. So those are the different ways amino acids should appear. Those all have the same correct stereochemistry. So to answer your question, why is asparagine a wedge? Because if we tilt our heads and sort of look at it from this perspective, nitrogen's on the left, carbonyl's on the right, alpha carbon is at a peak, and therefore it has to be a wedge. Okay, others? All right, well, the way to practice a newfound skill is to do it again. So we are going to do that again after the break. The peptide that we'll talk about is derived from a horseshoe crab, and it's antimicrobial. This actually kills bacteria. So we're going to draw it as a beta hairpin. It's got multiple cysteines. We'll talk about that after the break.